Welcome back to the deep dive. We are diving into, well, a stack of truly revolutionary research today. We're taking on what feels like the ultimate quest in modern medicine, which is moving past this old idea of disease as just an isolated mechanical failure, and finally seeing the human body for what it really is, a complex, interconnected, evolving ecosystem.
That shift, from the machine metaphor to the ecosystem, that is everything for the future of health. For decades, our success came from this very reductionist approach, right? We'd hunt for the single rogue gene or the one mutated pathway.
The one broken part.
Exactly. But to really master systemic disruptions, things like cancer, chronic disease, we need a holistic map. This is the whole philosophy behind what we call 4P medicine. So that's predictive, preventive, personalized, and participatory. But here's the catch. Embracing this holistic view, it generates such a staggering amount of complex, high-dimensional data that it just completely overwhelms human analysis.
Okay, so let's unpack that. The goal is to treat you, the patient, as this unique system that's constantly changing. So our mission for this deep dive is to understand the revolutionary AI infrastructure that's making this possible. We're talking specifically about these massive generative foundation models and specialized autoencoders. We really need to get into how they handle these huge data sets and how they actually drive diagnostic precision forward.
To map that systemic disruption, you have to start with the map itself, which means understanding the shift to what we call multi-omics. Think of it like you're mapping an entire city, not just one house. The genome, that's your architectural blueprint. It's the foundation, but it's static.
It's the potential, not the activity.
Exactly. Then you have the epigenomics, things like DNA methylation.
The annotations on the blueprint.
Precisely. The construction notes marking which streets are open, which buildings are active. Below that, you get the transcriptomics, the RNA messages. That's the dynamic real-time communication of what the crews are actually doing right now.
And then we get to the actual output, the machinery itself.
That's right. Proteomics and metabolomics, those are the protein machines and the chemical exhaust. They're the fingerprints of all the biological processes happening. And we can't forget the microbiome, this vast community of microbes living inside that city. They influence everything from traffic flow to power consumption.
So the challenge for you, the learner, becomes really clear really fast. Each ohm is a valuable perspective, but they're all kind of incomplete silos. To understand the whole system, you have to connect them. And the technical hurdle isn't just the volume of data, but the velocity and the variety. We're talking millions of data points per person across different timelines, different data types. I mean, traditional statistics just break at that scale.
That is exactly why AI has to step in as this master integrator. It's uniquely equipped not just to catalog all this data, but to find novel biomarkers and these complex risk signatures that only show up when you look across all these different layers at once. AI is what lets us build those dynamic, personalized health models for true prediction and prevention.
Okay, here is where it gets really interesting. Let's jump into one of the most exciting applications, liquid biopsies. These offer non-invasive disease detection. Our sources are focusing heavily on something called cell-free RNA, or cfRNA. This is stuff that's shed by cells, including tumors, right into the bloodstream. And a particularly strong class of these markers are orphan non-coding RNAs, or oncRNA.
oncRNAs are a fantastic target. Tumors actively secrete them at really high levels, so they give us this clear signal of the cancer cell state. The researchers have this beautiful description for them. They don't appear in isolation, but in these distinct patterns. They call it a constellation of stars in a starry sky.
That constellation analogy is perfect because it really captures the data challenge, right? If the signal's too sparse, if you only see a few stars here and there, you don't recognize the pattern, you miss the constellation.
That's the core problem. cfRNA data is incredibly sparse and noisy, especially in early-stage disease or when you can only draw a small amount of blood. This noise and sparsity, it just makes reliable detection, the kind you need for a clinical test, extremely difficult. That's why we need models that can fill in the gaps accurately.
And this brings us to XI1. It's this powerful, multimodal, transformer-based generative foundation model designed specifically to solve this high-noise, high-sparsity problem. Can you tell us why it's considered multimodal? Because that seems like a really critical distinction.
Right. Multimodality here means it's combining two different types of information about the RNA. It doesn't just look at the abundance data, how much RNA is floating in the blood. Crucially, it integrates that with RNA sequence embeddings. These derive from a totally separate model called RNA-FM. So it's merging the structural properties, the sequence of the RNA molecule itself, with its expression level. Looking at both structure and level together is just so much more powerful.
And the scale of this model is just immense. Pre-trained on over 306 billion tokens from a cohort of more than 13,000 blood samples. I mean, that's the kind of heavy lifting you need to learn these generalized biological rules.
And that scale is what lets it generalize. The architecture itself is designed for specific tasks. It uses methods like masked language modeling, which is how standard transformers learn context. But the real innovation here is these special tokens it learns to incorporate. Things like CD for cancer detection, TMO for tissue of origin, and BT for biofluid type.
So how does using a token like CD actually improve the outcome? What's it doing?
It lets the model contextualize the data it's seeing. So instead of treating every single sample the same, the model learns to filter its massive internal knowledge, specifically for patterns that are relevant to cancer when it sees that CD token. It turns it into a much more directed, context-aware prediction machine. And that drastically broadens its real-world use beyond just simple classification.
And in terms of performance, how good is it at actually filling in those gaps in the sparse data?
It's remarkably effective. The whole goal is robust reconstruction and denoising. When you compare Xi1's reconstructed values to the original, often noisy measurements, it achieved an R2 value of 0.89. To put that in perspective, the dataset average baseline for this kind of noisy data was only 0.57.
Wow. And that jump from 0.57 to 0.89 isn't just a statistical win. That fundamentally changes whether you can even use CFRNA in a clinical setting. It's making previously unusable, noisy data useful.
Exactly. And that leads to this huge practical benefit, cross-biofluid translation. The latent space it learns is so generalized that a classifier trained on, say, high-quality plasma samples can be reliably applied to serum samples. And serum is a biofluid that's far more commonly and easily archived in clinics worldwide. So Xi1 is solving a major practical logistics problem just by being a better denoiser.
Okay, so Xi1 gives us this incredible denoised, high-fidelity data from a single modality. But now we have all this complex, high-dimensional info pouring in from all the different omics layers. What do we do with the sheer scale of dimensions it creates? This is where we need tools to tame the curse of dimensionality. That's where autoencoders come into the picture.
Absolutely. Autoencoders, or AEs, are deep learning models that specialize in this exact task. They use this encoder-decoder structure. They take that high-dimensional input, say 20,000 genes, and they squeeze it through this low-dimensional bottleneck. This creates a compressed, nonlinear representation we call the latent space, and that latent space holds only the most critical information, which makes the data manageable.
And the framework for standardizing these tools is called AUTOENCODIX. It includes various architectures designed for this kind of multi-omics integration, but where it gets really sophisticated is in solving the biggest problem in deep learning, the black box.
Yes, the black box is just intolerable in medicine. You can't have it. So this framework introduces ONTIX, which is an ontology-based VAE, and it provides explainability by design. This is where the engineering really meets the biology.
Explainability by design sounds like a massive claim. How does constraining the model based on known biological knowledge, like reactome pathways, how does that actually deliver interpretability instead of just forcing the output into a pattern we already know?
It's actually ingenious. Instead of letting the decoder connect that compressed latent space to the full genome randomly, ONTIX restricts those connections. So for example, if a latent dimension corresponds to the immune process pathway in reactome, the decoder is only allowed to use the genes associated with that pathway when it reconstructs the original data.
So if that specific dimension lights up, we know exactly what biological process is driving the prediction. It gives the score context.
Precisely. It gives biological context directly to the prediction, which is so crucial for the personalized and participatory parts of 4P medicine. Using ONTIX, researchers found that latent dimensions tied to central nervous system cancers reflected high activity in very specific pathways, like immune processes and vesicle transport. This gives physicians actionable context, not just a black box risk score.
And beyond explainability, the AUTOENCODIX framework also has tools for data translation, right? For connecting those different omics layers. This is XModalix, the cross-modal VAE.
XModalix is the engine for systemic biology. It's really the core of it. It learns separate but strategically aligned latent spaces for entirely different types of data. Is asking, can we learn the common language that underlies, say, DNA methylation and gene expression?
And what were the most compelling results of this translation ability? What did it manage to do?
Well, the omics-to-omics translation is impressive on its own. It successfully translated DNA methylation data directly into reconstructed gene expression data. But the omics-to-image translation is the one that really makes you pause. XModalix used transcription factor proteome signature, so purely molecular data collected across different developmental time points to reconstruct images of C. elegans embryonic development.
Wait, hold on. It generated a picture of a developing organism purely from a molecular recipe. That is profound. It implies the model has genuinely decoded the underlying connection between gene activity and physical form.
It does. The source noted that even when the model was shown unseen developmental time points, the reconstructed images were still very close to the originals. This means the model understands the biological trajectory of development. It hasn't just memorized the training data. A critical detail here is the finesse needed in the training. You have to prioritize the reconstruction accuracy, or loss, while carefully weighting this class-based loss to make sure the different latent spaces align properly.
That brings us to the final piece of the puzzle. If we're going to feed these incredible AI tools, Xi1 and AUTOENCODIX, we need the absolute highest fidelity data we can get. We know that current single-cell RNA sequencing, cRNA-seq, suffers from high non-detection rates or dropouts, especially for transcripts that are lowly expressed but might be biologically crucial.
This dropout problem makes rare but critical biological events totally invisible. So to combat this, researchers developed RoTK and ROI. It stands for Robust Capture of Key Transcripts and Regions of Interest. It's a workflow that does the whole transcriptome analysis, or WTA, while simultaneously zeroing in on specific, difficult-to-detect targets.
How does it manage that? How does it do both functions without messing up the quality of the overall cell profile?
It uses two mechanisms. First, there's RoCK, which modifies the standard barcoded beads used in cRNA-seq. Standard methods rely on the poly-A tail of messenger RNA for capture. RoCK adds custom capture sequences to enrich for specific transcripts that might not have a reliable poly-A tail or are just at really low levels. Second, ROI-seq is basically spiking in specific primers during library generation to direct reads to a very specific region of interest, like a splice junction.
So you get the big picture from the untargeted whole transcriptome analysis, which tells you the cell's identity. And then you get the highly focused targeted data, which shows you the precise molecular event.
Exactly. And the key advantage the sources confirm is the detection rate. It achieved up to 99% positive cells for targeted transcripts without significantly compromising the quality of the overall WTA.
And the applications for this are immediate and clinically vital, I'd imagine, especially for detecting fusion transcripts in cancer.
Yes, the classic example is the BCR-ABL1 fusion transcript in leukemia. This fusion is notoriously hard to detect with standard cRNA-seq. The fusion junction is often too far from the 3' end, so the standard poly-A capture system just misses it. Using RoCK and ROI's targeted priming right across that fusion point, they successfully detected and distinguished between the minor E1A2 and the major E14A2 fusions in patient cells.
And that high-fidelity WTA data led to an unexpected discovery, too, didn't it?
It did. The concomitant WTA data, which identified all the other cell-type-specific RNA, it allowed them to detect that BCR-ABL1 fusion transcript in cell types they previously thought were unaffected, specifically in monocytes. This level of resolution dramatically changes how we see the disease landscape within a single patient.
That's the high resolution of molecular data, but systemic biology also demands context. We need to know where these molecules are within the tissue, which brings us to the final piece, the spatial dimension, provided by RoCK.
RoCK integrates spatial transcriptomics, which tells you where transcripts are located, with something called label-free vibrational biochemical imaging. It uses Raman microscopy. Essentially, it's decoding the spatial vibrational molecular architecture of tissues.
That phrase, vibrational molecular architecture, is so descriptive. What kind of information are they pulling out of that?
It lets them bridge specific transcriptomic programs, so what genes are active in a certain spot, with specific biochemical components, things like localized fatty acids, lipids, saccharides. They demonstrated this beautifully in studies of aging and wound repair in mice skin and lung. Basically, the Raman features themselves can be quantified and used as non-destructive biomarkers of cellular senescence or aging.
Which means we could potentially use this as a high-throughput screening tool, or even for in vivo monitoring of tissue health over time. I mean, that is the ultimate predictive and preventive vision of 4P medicine.
It is. We're truly witnessing what the source material calls oncology's Copernican transition. We're shifting from treating isolated molecular parts to appreciating the intricate, interconnected systems that define us. And these AI tools, Xi1 for denoising, AUTOENCODIX for integration and explainability, combined with high-fidelity data capture like RoCK and ROI, and the spatial context from RoCK, this is the essential infrastructure to finally realize that holistic, proactive vision.
So the key takeaway here really is the convergence. The future of personalized medicine isn't just about collecting more data, it's about integrating diverse, high-fidelity information. Combining cfRNA sequence information with expression levels, layering omics data with spatial images, analyzing the whole transcriptome alongside specific splice junctions. It's the meeting of all these disparate data layers that creates the real insight.
And that meeting raises a truly fascinating question, one that builds on that incredible work with X-Modalix. If our algorithms can successfully translate the language of one biological layer like DNA methylation or transcription factors into a completely different form, like gene expression data or even a physical image of a developing worm, what fundamental unseen codes of health and disease are we on the verge of decoding? And how will that ability to translate biology change our understanding of life itself?